When you're doing molecular cloning and using antibiotic-based selection, this is the type of plate that you want to see when you come in the next morning. And so yay to Karina for getting these nice colonies. So each of these little clumps represents a bacterium that kind of plopped down and started making copies of itself. And so you get these clumps of bacteria that are genetically identical. And they're containing the plasmid that you put in them. And so the plasmid that you put in them has a gene for making the protein you want them to make, but it also has a gene for making a protein that is going to allow them to survive in the presence of the antibiotic. And so each of these kind of gloopy dots is what we call a colony. And so in these plates, we have LB agar, which is basically bacterial food in a gel, and then it also has kanamycin, so the antibiotic that the bacterium that have the plasmid will be resistant to. And so the bacteria that actually took in that plasmid will now kind of plop down on the plate, and they will start growing, and they'll make copies of themselves upon themselves. And so you get these kind of gloopy dots called colonies, and you want to see them nice and spaced apart and kind of big. Sometimes you might want to plate different volumes, and so this is going to then allow you to make it so that you have these isolated colonies as opposed to regions where things are kind of really just, the colonies are really close together and overgrown. Because although the bacterium hopefully have the plasmid that you want to put in there, it could also be possible that they have the plasmid that was the template for making mutations to the plasmid if you're doing like site-directed mutagenesis, or you're doing some other PCR-based method to do your cloning. The template plasmid will also have the antibiotic resistance gene and therefore also be able to grow. And so you want to isolate single colonies and actually send them for sequencing to make sure that it is the version that you want and that there aren't any errors elsewhere in the plasmid. And this is a good start though, is having colonies to go and actually be able to sequence. Now to sequence them, you typically grow them up in a larger culture, and so you take like a pipette tip or a sterile toothpick and you just boop, and you put them in like 5 mils of LB, let them shake overnight, and then you're going to use like a mini prep kit or some other alkaline lysis kit in order to purify the plasmid out of these cells. And then you could go and send it for sequencing. It used to be that you had to like send it for sequencing, and like if you wanted the whole plasmid sequence, you had to do like in multiple primer pairs, so that because they would do kind of primer based methods where you just amplify specific regions of DNA, and then the reads weren't long enough for you to go all the way around. But now there are whole plasmid sequencing options where they'll sequence the whole plasmid, and you don't even need to give them primers, and it's not really that much more expensive, especially if you wanted to sequence the entire plasmid. It's like a cheaper option. Easier, cheaper, we go for that. Unless I'm having problems with the cloning, I typically just start by sending it for sequencing. But there are other methods that you can do to kind of get a check before you send things as to whether it likely has your mutation. This includes colony PCR, where you kind of take a little bit of the colony and you do PCR directly on it without having to purify out the plasmid, and you design primers that are only going to give you a product, or only going to give you a specific product if you have the mutation present. The problem with this is that it's still not giving you the entire plasmid, and you could have mutations elsewhere that you don't realize because you are only seeing that the mutation you want is present. But this can help you kind of decide which colonies are worth sending for sequencing. But then I would still send the entire plasmid for sequencing to get that kind of better information. You can also use analytical restriction digest, where basically if you introduce like a silent mutation or something, or somehow the mutation introduces a restriction enzyme cut site, you can then purify out the plasmid and do a restriction digest to see if you get the cut pieces that you're expecting. But with that, you have to actually purify out the plasmid. Sequencing these days is just really easy, and as long as your cloning efficiency is good and you're getting good results, then you can just kind of choose a couple of the clones and send them for sequencing. And then you don't have to worry about buying primers to test whether the cloning worked. And so we're going to do whole plasmid sequencing, but we're going to test a few different clones because we want to make sure that we're getting a clone that actually has the version of the gene that we want and doesn't have mutations elsewhere in the plasmid. If the colonies are too close together, then you're not going to be able to isolate single ones, and so you might get back a sequence that's kind of a mix of multiple different sequences, like one of the good one and one of the bad one or something like that. And so you want the colonies to be nice and big and isolated. You don't want to choose a little tiny colony on the side of the bigger colony that's like a satellite colony. This is especially common with ampicillin resistance, because ampicillin actually, the resistance protein for it gets shipped out of the cells and kind of protects the cells around it, so you can get these tiny little satellite colonies. And so you can avoid that by taking the plates out of the incubator sooner and things, but you want to avoid taking those little satellite colonies. But having colonies in the first place is a really good start, because sometimes you don't get colonies and it's sad. But when you don't get colonies, sometimes you can trick yourself into thinking that they're colonies, but it's really just like a bubble in the plate or something. When you're trying to look and see if things are colonies without opening up the plate, you can kind of like look on the side and see if you see little bumps. And because the bacteria are going to grow on the surface of the agar, and then they're going to kind of like clump on themselves. And so that is the basics of what you want to look for when you go and take your plates out of that incubator when you're doing this antibiotic-based selection, is these nice clumpy dots, and we call those clumpy dots colonies. And each of those colonies should, at least theoretically, be genetically identical, but they might be different from one another, but all the bacteria in that colony should be identical. But you need to actually go and sequence the DNA out of there in order to know if the sequence is correct. And so right now our DNA, our plasmid, is in DH5 alpha strain. And so DH5 alpha is a strain of bacteria that's really good at making a lot of copies of DNA and doing it with a high fidelity. So when we say high fidelity, we're basically meaning that it can do a lot of this copying without making mistakes. And so you might hear high fidelity polymerases, like with your PCR, they don't make mistakes, or they make fewer mistakes, and often they have like a proofreading domain thing. With high fidelities in these bacteria, basically they're able to make a lot of copies of their DNA and not make errors. And then basically they're not good at making protein though, and so if we want to make protein, we got to stick them in another type of bacterium. And so we typically use like BL21, DE3 cells for doing the actual expression. But for the transformation initially, when we're doing the cloning stuff, we put them into cloning cells like this DH5 alpha, which are high fidelity, but they don't have the bells and whistles for making the proteins that we want to make. And so we don't care as much about fidelity in the case when we're just trying to make the protein, but we do care about it when we're making lots of copies of the plasmid. And we need to make lots of copies of the plasmid in order so that we can sequence the plasmid, make sure it's okay, and so we can get enough of the plasmid so we can do things like stick it into the other bacteria in order to have it make protein for us. So when we talk about cloning cells versus expression cells, that's what we're talking about.